Hi, my name is Ameer Taha. I'm a professor in the Department of Food Science and Technology at the University of California, Davis. The title of my talk today is The Role of Omega-6 Linoleic Acid and its Oxidized Metabolites in the Brain. Before I start, I want to thank Zero Acre Farms for the invitation to present on some of our recent work on this topic. I'm going to give a brief background on the all the science leading to the research questions we've been trying to deal with over the past couple of years. Then I'm going to dive into the metabolism of linoleic acid and its oxidized metabolites, or also known as OXLAMs, oxidized linoleic acid metabolites. And I'm going to wrap up by addressing their role in the brain. So let's get started with some background information and some of our questions. A crash course in chemistry. Linoleic acid, or LA, it's a nutritionally essential omega-6 polyunsaturated fatty acid found in the diet. So let's dissect this, some of the terms in this phrase. It's nutritionally essential because unlike plants, we cannot make it endogenously. So we need to get it from the diet, and plants are the best source for LA. We usually obtain it from dense sources or seed or plant oils, which I will talk about in the next slide. And it's an omega-6 fatty acid, meaning that if you look at the molecule, this is linoleic acid, this is the acidic terminal, and this is the terminus of the molecule, or the terminal of the molecule. Omega-6 simply means that the first double bond, so as you can see, it has two pairs of double bonds. Omega-6 means that the first double bond starts on the sixth carbon from the terminus of the molecule. This is unlike omega-3 polyunsaturated fatty acids, which I'm sure you've seen on, in CVS or other drugstores, where the first double bond starts from the third carbon of the molecule. So 1, 2, 3. Omega-6, you have the first double bond on the sixth position. And such subtle differences really make a night and day difference in the properties of these molecules. Lastly, it's polyunsaturated, meaning that it has more than one double bond. So both omega-6 and omega-3 fatty acids are polyunsaturated because they have more than one double bond in their structure. In the case of omega-6 linoleic acid, it has two double bonds. For alpha-linolenic acid, or ALA, which I haven't shown the structure here, it has three double bonds. Therefore, both of them are polyunsaturated fatty acids. So where do you find LA? And I've also given you sources of ALA. Mainly what they call erroneously vegetable oils. Of course, olives are not vegetables. Olives are fruit. So you can find them in fruit or mainly plant and seed oils. So specifically sunflower, corn, and soybean oil are generally high sources of LA, and less so would be olive oil and canola oil. You can appreciate that sunflower oil has approximately 75% LA. So let's talk a little bit about how this was discovered and how the fields opened over almost close to 100 years ago. So in the 1930s, George Oswald Burr and his wife Mildred Burr, they discovered that omega-6 LA was one of two essential fatty acids. 2 years later, they discovered that the other essential one was alpha-linolenic acid, or ALA. They showed this by demonstrating that LA is needed to optimize growth in rats. So as you can see on this right figure, it's a busy slide taken from their 1930 paper. But as you can see on this right figure, oils such as corn oil or linseed oil, which contained LA, resulted in optimal weight gain in animals that were otherwise deficient in LA. And this led them to conclude that LA, or linoleic acid, and possibly other fatty acids are essential fatty acids in the diet. Of course, they discovered 2 years later that the other possible fatty acid that's essential as well is ALA, or alpha-linolenic acid. Today's talk is going to focus more on LA, linoleic acid, omega-6 LA. The evidence for their essentiality was further established in humans by Arild Hansen, Dr. Burr's student. So as you can see in this figure, this child on the left here is actually deficient in LA, and you can see that growth is stunted. And also, unfortunately, the child suffered from skin lesions. On the other hand, on the right side, this child was initially deficient, but then LA was reintroduced in the diet. And in doing so, growth became normal, and the skin lesions disappeared. So you can start to see that the dermatitis started to go away. And the authors concluded that LA is required at 1 to 2% energy of calories, of daily calories. Of course, in adults, because adults are not growing, the estimated requirements are probably around 0.5% energy. But this hasn't been experimentally established just because it's hard to deplete linoleic acid from adults. It's really everywhere in these plant oils that I've shown you earlier. But we can assume the requirements are somewhere between 0.5 to 2% energy. I probably cheer more on the 0.5%, but I'm sure one can put forth an argument that it may be around 2%. In any case, our current intake levels are much higher than 0.5 and 2%. They're at approximately 7% energy, so at least threefold the minimum requirements. Now, this wasn't always historically the case. So if you look at this figure on the left side, you can see, so just look at the top graph here, where on the y-axis you have the percent LA intake, and on the x-axis is time, historic levels of consumption. So you can see that in the early 1900s, LA was at about 2% energy consumption level. But starting in the 1940s or so, its intake started to increase with the advent of soybean agriculture and soybean agriculture, from which you would get the oil, as well as corn agriculture. So LA levels in the US at least, and then most of the world started to increase significantly, reaching about 7%, 7.2% by the late 1900s. I know it's 2021, so is there more recent data? Yes. So we have shown actually we've shown that using enhanced data, that most of the population in the US is still at the 7% energy mark, which is here. But the significant amount of adults in the US are actually also above 10%, I think around 20% of the population. So LA is still an abundant source of calories, or it's quite abundant in the diet. So what's the problem here? LA is actually a precursor to pro-inflammatory OXLAMs, or oxidized linoleic acid metabolites, which can be made either through enzymes or by non-enzymatic processes, or what's called as autooxidation. The best example of autooxidation is when you cook with oil. You oxidize it, and if you're using a high LA oil, it gets oxidized, and it can form some of these OXLAMs, oxidized linoleic acid metabolites. Our bodies have a lot of enzymes that actually spend a significant amount of energy oxidizing LA to make these OXLAMs, and for a purpose. So they are important at some level, but in excess, they can be pro-inflammatory, at least when made in vivo, so in our bodies, different from when it's made on a cooking pan. And this is just a complicated diagram of the pathways. So you have linoleic acid, you have lipoxygenase or cytochrome P450 and other enzymes, simply adding an oxygen to the molecule in different locations, in different forms, to yield these OXLAMs. So oxidation really means you're adding an oxygen to the molecule. So as you can see here, you're adding a hydroperoxy group, OOH. Here it's an OH. Here you're adding an epoxide, this oxygen hat-like structure. So you end up with a diverse set of oxidized linoleic acid metabolites with different properties and functions. Now, in 2017, we found that OXLAMs make up the majority, up to around 60% of oxidized fatty acids consumed in the US diet. But I just want to point out that the levels, absolute concentrations, are much lower compared to how much LA we get in the diet. What does this mean? Oh, sorry. So in our estimates, we concluded that per day in the US, we get about 0.64 milligrams of OXLAMs per day. And the total amount of oxidized fatty acids, also known as oxylipins, is about 1 milligram. Compared to LA, we get about 16 grams of LA. So the units matter here. 16 grams of LA per day versus 0.6 milligrams of OXLAMs per day. So even though OXLAMs make up the majority of oxidized fatty acids we consume, the amount we get is less than a milligram. It's minuscule compared to how much LA is in the diet. And again, LA is at least 7% of total calories in the diet. But back in the 50s, Dam and colleagues, they found that LA induces encephalomalacia in chicks fed a vitamin E deficient diet. So what they did was they simply added LA to the diet of chicks or chickens. They removed vitamin E, and when they removed the vitamin E, they found that they had significant neurological problems, so characterized by neurodegeneration and ataxia, so imbalance basically, and some actually died. And they hypothesized, and some papers showed later, that this was due to the auto-oxidation of LA in the diet. So, like I said earlier, LA can make OXLAMs by non-enzymatic oxidation, just like when you fry with oil. So in chickens, when this happens, it actually causes brain problems and neurological problems. So this led us to ask some key questions. First, what about mammals? We're not chickens. We're mammals. So the first question was, are dietary OXLAMs bioavailable in mammals? Do they get to the brain? And what do they do in the brain if they do get into the brain? So again, let's take a step back, or 2 steps back. LA is the most abundant fatty acid in the diet. It's a precursor to OXLAMs, which can be formed in your frying pan or enzymatically by enzymes in our bodies. But we take a lot less OXLAMs compared to LA. Yet there was this paper in the 50s showing that OXLAMs in the diet, even though they're much less compared to LA itself, it caused encephalomalacia in chicks. So we wanted to see if these findings extend to mammals. So we started with rats to check this out, basically. So this is going to lead me to the next topic of my lecture on the metabolism of LA and its OXLAMs to address the bioavailability and brain entry questions. And then the role in brain will deal with the brain effects question. So these are the 3 questions we laid out. Are OXLAMs bioavailable? Do they get to the brain? And what the hell do they do in the brain if they get to the brain? So let's talk about how LA and its oxidized metabolites are handled by the body. So what we did, we sort of did a replica of that chicken study in mice. We fed them a low LA diet, a high LA diet, or a diet that's high in OXLAMs. So these are, so if you take your eyes to the bottom of this big table, you'll realize that the OXLAMs in the low and high LA diets are pretty low, but in the high OXLAM diet, they're tenfold compared to the low OXLAM diet. So let's see if I can get my mouse. So 200 to 259. LA was, again, this is milligram per gram of diet. LA in the low LA diet was around 28. In the high, it was 3 times as much. In the OXLAM diet, it was low in LA, but it was supplemented with these OXLAMs. So 3 diets were fed to the mice for 8 weeks. And we looked at the incorporation of OXLAMs in liver and then brain. We just wanted to know, do they get into the body? If you feed them through this dietary intervention, the high OXLAM diet, do they accumulate in liver and brain? So what we found was the high LA. So these are all the OXLAMs, so don't mind the abbreviations. Like I said, there's so many OXLAMs that are formed in the diet. And these are, to the right is metabolites of other polyunsaturated fatty acids that we looked at, arachidonic acid and dihomogamma-linolenic acid. So let's ignore these for now, and let's focus on the LA-derived oxylipins, which are the OXLAMs. So as you can see, and the y-axis depicts concentrations. So this is the low LA group, and the middle bar is the high LA group, taking this OXLAM as an example. So when you increase LA in the diet, you can see that the liver OXLAMs increase. But when you feed the mice OXLAMs, their liver OXLAMs don't change compared to the low LA diet, the control. And this is true for all the OXLAMs that we looked for. Again, high LA increases OXLAMs. OXLAMs themselves in the diet don't really change liver OXLAM concentrations. We see a similar thing in the brain. Again, where the high LA diet increases the OXLAMs, but the OXLAM diet does not move them. It doesn't change their concentrations. So what does this mean? It means that feeding a high LA diet increases tissue OXLAM concentrations, and that's thought to be done enzymatically. So you take LA through your diet, and your enzymes oxidize it to make OXLAMs, which accumulate in tissues, liver and brain. But feeding OXLAMs doesn't alter OXLAM tissue concentrations. So the question is why? There's 2 possibilities. Either the OXLAMs are not absorbed in the first place, so you have them in the diet and they just don't get absorbed, they exit the other way. Or the other possibility is that they get in and they get metabolized quickly. So we're not able to measure accumulation in liver or brain or other tissues over time. To test this question, what we did was we gave an OXLAM tracer. So we use here deuterated D4, which is a label that is added to an OXLAM. In this case, 13-hydroxyoctadecadienoic acid, or 13-HOD. We chose this OXLAM because in our studies we found that it's the most abundant one in the diet. In oils, in French fries, this is the most abundant one. So we gave it to the rats by gavage or IV, and we looked at tracer incorporation in liver, heart, adipose, and brain using mass spectrometry. So what we found was actually interesting. So if you take your eyes with me to the JN, so JN, we did some kinetics to figure out the incorporation coefficient, rates, turnover, etc. What matters in this slide is the JN. JN is what actually gets into tissues. So what we found is that the 13-HOD does not get into the brain, at least 90 minutes after injecting or gavaging. But it does get into adipose tissue, liver, and heart. And what was interesting is that it loves adipose tissue. A lot of it gets into adipose tissue compared to liver and even heart. So this showed us that OXLAMs get into, so you give them to the animals, you gavage them, they get into the body, and once they're in the body, they distribute to tissues, peripheral tissues, except the brain. Now we also found that the turnover. So if I bring your attention to the lowest part of this table, the half-life coming from the turnover is pretty short. So once they get into tissues, they only stay there for a couple of days. For adipose, it's less than a day, 0.3, a third of a day. For liver and heart, it's about 3 days. What this means is that they're being turned over quickly. And this is probably why we were not able to see changes in the liver or the brain when we fed OXLAMs to the mice. Just because once they're absorbed, they get turned over rapidly. And we're not sure what they're being turned over into at this point, to be honest. But there's something happening there where they get into tissues and they get rapidly kicked out of tissues or metabolized into something else that we haven't identified. So what does this mean? It simply means that dietary OXLAMs, which are the most abundant oxalipins in food, as I showed you before, are absorbed and they're incorporated into peripheral tissues, but not the brain. So why didn't we see it in the brain? And you can see our frustration at that point by the double question mark and then the exclamation mark, where I indicate that when we measure brain OXLAMs in rodents, we see them there. Yet when we inject a tracer OXLAM, it doesn't get into the brain. So how is the brain getting its OXLAMs? So one possible explanation is that LA itself may be the primary source of OXLAMs in the brain. And this sort of leans on some of what we found in the prior mouse study, where when we gave LA, when we increased the dietary composition of LA, the OXLAMs in the brain and the liver increased, suggesting that they took the LA and enzymatically converted them into OXLAMs. And there's evidence to show that increasing dietary LA regulates brain OXLAM concentration. So this slide on the y-axis is the concentration of the OXLAMs we measured. Again, 9-hod, 13-hod, these are all different types of OXLAMs. Don't mind the abbreviations. But what this shows is that as you increase dietary LA beyond 5% energy, so the white is 0.4 energy, light gray is 5%, and dark gray is 10%. As you increase dietary OXLAM concentrations, the brain OXLAMs increase. You reach a ceiling effect, it appears, between 5 to 10%, so you don't get that much of an increase compared to between 5 and 10% compared to 0.4 relative to 0.4%. But again, this means that what it's showing is that more LA in the diet means higher OXLAMs in the brain to a certain point. So this led us to test whether LA is converted into OXLAMs in the brain and by the brain itself. To do this, we fed rats a graded LA diet, so a low, medium, high LA diet, and we infused with labeled LA, C13 LA, and we measured unlabeled and labeled OXLAM concentrations in plasma and brain. So just to go over the diet quickly, we fed a low LA diet, 0.4%, sort of what we call an adequate LA diet, or medium, 5% energy. So this is close to what we consume here in the US, roughly 7%, so 5 is close to 7, and a 10% LA diet, which sort of resembles what a relatively high LA diet, basically. So what we found was that, and I'm going to take your attention to the right graph here. What we found is that if you increase dietary LA, you actually increase the turnover of LA into OXLAMs. At least we picked one of them, which is 9-hod, C13 9-hod. So this is a 0.4% LA diet, 5% and 10%. What this is showing is two things. One is that LA entering the brain gets converted into OXLAMs. It gets turned over into OXLAMs, and that this turnover is pushed or is enhanced when dietary LA levels increase up to a certain point. So you get this ceiling effect I was talking about beyond 5% LA. So these results tell us that dietary LA is the main source of brain OXLAMs, not dietary OXLAMs. LA entering the brain is rapidly metabolized into OXLAMs, and increasing LA pushes this metabolism further, so it leads you to convert more OXLAMs by the brain. So this basically covers the metabolism of LA and its OXLAMs in vivo. And I'm going to jump now into the role of LA and OXLAMs in the brain. We started these studies by simply addressing basic physiological questions. Our first question was, do OXLAMs regulate neurotransmission or brain signaling, in simple terms? And what we found, if I can take your attention to the top right graph here, is that similar to arachidonic acid-derived prostaglandin E2, this may sound jargon to you, but this is a widely studied molecule, PGE2. It's actually also sold as a drug to induce labor. So PGE2 has been shown previously to regulate brain signaling. So we confirmed that in this slide here, in this graph. So this is vehicle before adding PGE2, and this is after adding PGE2. So it increases what we call here PERT pulse facilitation, so neurotransmission. What we found in this study is that 13-hode, an OXLAM or a metabolite of linoleic acid, also did the same thing compared to PGE2. So 13-hode also increased neurotransmission similar to PGE2 compared to vehicle. LA itself didn't do much, and arachidonic acid, the precursor to PGE2, didn't do much either. And there was a dose-dependent effect. The bottom line here, the punchline, is that this is the first study to show that 13-hode, or one of the OXLAMs that's abundant in the diet, are actually involved in neurotransmission. We also moved to cell culture studies with Dr. Pam Line at UC Davis, and showed that these OXLAMs are also involved in regulating neuronal morphology in developing rats. And they do so in a sex-specific manner. So what you see here is dendritic properties, including dendritic length, branch points, and tips. So if you think of a neuron, it has a cell body, it has dendrites. So dendrites are like the antennas that are receiving signals, and then they pass them into the cell body, and the cell body shoots them through axons into other, so it sends the message to other neurons. What we're finding is that these dendrites are impacted by these OXLAMs. For example, 9-hode here seems to increase dendritic dendrite length in males only, but not females, and at a certain dose of sorry, I lost my pointer, at a certain dose of 100 nanomolar. In contrast, other OXLAMs like 9-oxode and 12-13-EPO decrease dendritic length and branching points in both males and females, also at certain doses. What this is telling us is that these OXLAMs play a significant role in neurodevelopment because they regulate the architecture of neurons in early life. For good or worse, it's hard to tell at this point. We're doing follow-up studies to really answer that question. But for now, at least we know that they're regulating neuronal morphogenesis or neuronal architecture by affecting the dendrites, or these antennas that receive messages from other neurons. And in adult rats, we're showing that they regulate the packaging of a signal or neurotransmission. We followed these studies with in vivo studies, so in the animals themselves. We wanted to know whether they are involved in regulating the response to ischemic brain injury. So ischemia is an excitotoxic state, and it occurs, for example, after a stroke, an ischemic stroke, and it's very damaging to the brain. So here we showed for the first time that in different brain regions in the rat, these OXLAMs on the x-axis, all the different OXLAMs, the y-axis is their concentrations, and you have different brain regions: cortex, cerebellum, hippocampus, brainstem. we're finding that these OXLAMs are massively produced during ischemia compared to controls, non-ischemic controls, suggesting that they're potentially involved in regulating this response. And they may therefore serve as a target for mitigating the effects of ischemia that is damaging to the brain. And we also looked at inflammation, neuroinflammation in rats. Here, what we did was we gave rats a low LA diet, so 0.4% LA, or an adequate LA diet, 5%, so similar to, or 7% in humans. And we challenged them with LPS by injecting bacterial lipopolysaccharide into the brain, or artificial cerebrospinal fluid, which is a vehicle. And what you see is when you inject lipopolysaccharide into the brain of rats maintained for 15 weeks on the 5.2% energy LA diet, you see an increase in prostaglandin E2, which is an inflammatory marker. So you see an increase in inflammation. But this increase was blunted or almost abolished when the rats were on a low LA diet, a 0.4% LA diet. And COX cyclooxygenase is the enzyme that makes prostaglandin E2. So this confirms that with the LPS, you increase cyclooxygenase activity, leading to more prostaglandin E2. But this activation of this enzyme is blocked when you maintain the rats on a low LA diet. And we're still working on this, but we're thinking that these inflammatory effects are mediated by the OXLAMs, which are pro-inflammatory. So when you remove or reduce LA from the diet, you reduce OXLAMs, which is why you're probably not getting this inflammatory response you see in the 5.2% LA dietary group. So all in all, the results indicate that OXLAMs are involved in regulating an abundance of processes in the brain: neurotransmission, neuronal morphogenesis, the response to ischemic brain injury, and also neuroinflammation. And this is what our work highlights. We worked with Dr. Chris Ramsden at the NIH to, of course, try to move these findings to clinically to establish some clinical relevance. So in humans, we reduced LA intake in people with chronic migraines. So migraine is an inflammatory condition, as you know. And we tested whether a diet low in LA, with or without fish intake, reduces headache frequency. Fatty fish is a source of omega-3 fatty acids, which include ALA that I've mentioned many slides ago, but also the longer chain omega-3 fatty acids, eicosapentaenoic acid, EPA, and DHA, which have known anti-inflammatory effects in vivo. So we wanted to know if the combination of lowering LA and increasing omega-3 fatty acid intake can modulate headache outcomes. These are the diets that were used by Dr. Ramsden. So there were two groups, the low N6 and the high N3 low N6. And the cohort consisted of patients with migraine that were given the low N6 diet for 12 weeks, or the high N3 low N6 or H3L6 diet for also 12 weeks. And we compared migraines before and after, basically. So here in the L6 group, or low N6, LA was reduced from 7.4% energy to about 2.4% energy. In the H3L6 group, it was reduced by similar amount, but EP and DHA were increased from 47 milligrams to about 1.5 grams per day. And this was achieved by really substituting high LA cooking oils, the ones I mentioned before, soybean, safflower, etc., with coconut oil, low LA olive oil, macadamia nut oil, and flaxseed oil. And the fish intake and the EPA DHA came from increasing fish intake, fatty fish intake. So in that study, it was observed that the L6 diet sort of reduced headache frequencies and severities in the groups, but it was non-significant. but the H3L6 diet significantly reduced headache frequency, headache days per month, headache hours per day, as well as severity. And this was significant compared to the L6 diet, suggesting that LA lowering combined with increased omega-3 fatty acid intake can potentially reduce migraine frequency in these patients, likely through modulating the inflammatory by modulating OXLAM levels in vivo. There was also an association between LA levels in the blood and headache hours per day. So you can see that the highest association was between the highest predictor for headache severity was LA, circulating or plasma LA levels, basically, followed by omega-3 EPA, DHA, and then finally arachidonic acid, which is a elongation desaturation product of LA. So these findings tell us that LA lowering is anti-inflammatory because it reduces headache frequency in patients with migraine, especially when combined with EPA and DHA. So this is the final slide. All in all, our results or the take-home message for you is that the brain derives its OXLAMs, its oxidized linoleic acid metabolites, from dietary LA, not from dietary OXLAMs. LA entering the brain is rapidly converted into OXLAMs that regulate inflammation, neurotransmission, neuronal morphogenesis, and also the response to ischemic brain injury. LA lowering may enhance, at least in humans, it appears to enhance the benefits of omega-3 intake in inflammatory disease, in this particular case, migraines. Of course, much more work needs to be done to address whether this is a broad spectrum effect on other inflammatory diseases. With that, I'm going to wrap up by thanking members of my lab who contributed to these projects or are contributing until this day, as well as the funding that supported a lot of this work. And I thank you so much for your attention. Have a great day.